Welcome everyone. And firstly, thank you so much for your nice and kind efforts for the coordination for this oral presentation. I am Assistant Prof. Dr. Hayder Al-Mossa from the Hayder Institute of Fertility Diagnosis and Assistant to Predictive Technologies at An-Nahyan University from Iraq. My oral presentation entitled The Evaluation of the Clinical Role of Testis Expressed Protein 101 and Extracellular Matrix Protein 1 as novel predictive markers in relevance to the testicular sperm retrieval and differentiation of obstructive from non-obstructive Azoospermia. First of all, we want to know the Azoospermia. So before that, we want to know the infertility. The infertility is the inability to achieve a pregnancy within 12 months of a regular unprotected intercourse. The main infertility was ranged from oligospermia to azoospermia. The azoospermia, which can affect about 100% of the male population and up to the 20% of male infertility situations. So the azoospermia is the absence of a sperm in the seminal fluid and has two major types. So it's characterized mainly by zero sperm count. It can be divided into two major types, obstructive azoospermia and non-obstructive azoospermia. The latter type, which can be divided histologically into hyperspermatogenesis, maturation arrest, and Sertoli-only syndrome. The degree of spermatogenesis and the success of sperm recovery, all of these, in addition to the testicular biopsy, histopathology, and the sperm retrieval technique, all of these are the main basis of diagnosis and the treatment of azoospermia type. These are invasive procedures and have surgical and anesthetic complications and also the cost. So we need an alternative, non-invasive procedures which work urgently to differentiate between the types of azoospermia. Seminal plasma, which can be originated from the seminal vesicles as 65% and prostate 25% and the testis epididymis to 10%. The periurethral glands, which is the minimal contribution from them. The seminal plasma is rich with epididymal testicular proteins and directly participate in the sperm production and the maturation, or also the interaction with the zona pellucidum and even the fusion with the oocytes. They are not available in other biological fluid like the blood due to the presence of blood testis and the blood epididymal barriers. The semen and the seminal plasma remain the single available fluids for the non-surgical diagnosis of male infertility. The new research in the subject's proteomics, which mainly depend on the seminal plasma proteins, gives a promise for advancement of the novel male infertility biomarkers, which the TESE 101 and ECM1 is the major or one of these biomarkers. The seminal plasma protein-based assessment, TESE 101 and ECM1 are already discovered and under final development for the clinical use. The main principle of their assessment is the immunoassays, which could be used in the diagnosis of azoospermia types, prediction of sperm retrieval procedure outcome, and decrease the overall cost of the azoospermic patient diagnosis. The testis expressed protein 101 protein can be coded by the testis expressed 101 gene in mice. In human, this gene located on the long arm of chromosome 19 and present in testicular germ cells tissue. Its cleavage and detached from the cell surface of epididymal sperm as it passes throughout the head of the epididymis. The main functions of this marker, it can be involved in the spermatogenesis. Previous studies on the mouse, they found that the mice with TESE 101 had normal mating capability and they are unable to give birth to osteoporosis. And in epididymal sperm maturation, it's involved in the glycosyl phosphatidylinositol and proteins activation and the cleavage for permitting sperm donor plasmid binding. Other functions relevant to sperm-egg interaction. The TEX101 regulates the ADAM3 function, and lack of TEX101 was associated with a fertile phenotype, which is the main defect in the sperm movement into the oviduct. And TEX101 is cleaved and binding to the communal cell surface, causes calcium movement, also protection of progesterone from communals to encourage the communal layer penetration. The new second marker, which is the extracellular matrix 14-1, that's encoded by the ACM1 gene, is extracellular matrix 14 that maintains the integrity of tissue for the interaction with variety of structural and extracellular proteins. It will play an important role in extracellular matrix formation, cell adhesion, cell signaling, and regulation of tissue differentiation and or maturation. The main expression of it is widely in many tissues like liver, GIT, like small intestines, lung, ovary, prostate, testis, and adnexal epithelia, including the hair follicles and the sweaty glands. Within the epididymal tissue, here a high level of expression will be exhibited within it. The main functions of this marker, the exact biological function of it, is not elucidated yet. The ACM1 receptors facilitate the stem cells to connect to the local microenvironment. The seminiferous tubules is necessary for cell renewal, proliferation, differentiation, and survival of spermatogonia. All these findings suggest that germ cells of idiopathic non-obstructive azoospermia individual are lost ability to bind to the basement membrane of seminiferous tubules. The extracellular matrix 1 plays a significant role in regulating spermatogenesis, particularly spermatogonia and Sertoli cells and the blood Testis barrier. The main objectives of this study or this topic is to estimate the clinical role of seminal plasma proteins, TEX101 and extracellular matrix 1 according to the type of azoospermia, assess the predictive rate of seminal plasma protein and relevant to the sperm retrieval technique, the testicular biopsy. And the third aim or objective is the diagnosis of non-obstructive and obstructive azoospermia and the subtype of non-obstructive azoospermia, which here not discussed, just the major lines of them. Patients, materials and methods. History, which include medical, surgery, and infertility, physical examination, and investigations like the seminal fluid analysis, post-ejaculation urine examination, hormonal assay of the reproductive hormone like follicle-stimulating hormone, luteinizing hormone, testosterone, seminal plasma proteins, TEX101 and ACM1, and the testicular biopsy. The materials, different devices and instruments and equipment like the vertical plastic media, the kits for testosterone, follicle-stimulating hormone and luteinizing hormone will depend upon them to diagnose the azoospermia and obstructive azoospermia in relevance and in addition to other factors, human TEX101 ELISA kits and human ACM1 ELISA kits, operative procedures which include many instruments and equipment, and testicular biopsy. Here the study design. The study includes 65 azoospermic subjects. We collect the seminal plasma proteins, hormones, testicular biopsy, and relevance to the sperm retrieval, negative or positive, and see the markers we studied them in relevance to these main patterns or parameters. The materials here, the ELISA kits, as I mentioned before. Here the vertical plastic media. The steps of testicular sperm extraction here can be summarized in this or these images. Results. Here the characteristics of the patient. The sample size, as I mentioned before, is 65. The mean age of them, 33.37 plus minus 6.99. The duration, the median here is 7. The primary fertility was here 60 cases and 5 cases are secondary. Type of Azoospermia, the obstructive type was 10, and the major number of cases was the non-obstructive Azoospermia, which is 55 here. This table can summarize the spermatotypal rate according to the type of Azoospermia. The positive retrieval was 36, obstructive Azoospermia here 10, and the 26 is the non-obstructive Azoospermia. The negative retrieval here is the totally 29, 0 in obstructive and 29 non-obstructive. Here the p-value was significant. The ECM1 and TEX101 parameters in Azoospermia men. The ECM1 was 530, and this is the total number, 65. And non-obstructive, with 10 cases, was 469, and non-obstructive was elevated as 1,629. Here the comparison and the difference here is significant, highly significant. The text 101 has totally 1.05, 1.222 in the obstructive and 1.444 in the non-obstructive. Also here an elevation here found in the non-obstructive type. Here there is a high significance. Characteristics of work analysis. Here the sensitivity and specificity parameters. The cutoff values in the ECM1 was more than 943. In text 101, more than 0.79. And the accuracy here was 87.1, and in text 101 was 76.9. And the sensitivity here was more in ECM1, which is 87.3, and in text 101, 61.8. The specificity was 90% for the both parameters. And this table summarize the ECM1 and text 101 according to spermatotypal outcome in non-obstructive Azoospermia. Here, as we mentioned, the positive spermatotypal was 26 and the negative was 29. The ECM1 was in the positive as 1,492 and the negative was 1,613. Here the comparison was not significant, and the text 101 here, which was more in the positive spermatotypal, and here negative was 0.31, and here the significant comparison was significant difference between these two groups, positive and negative spermatotypal. The cutoff values here for text 101 are 0.92 cutoff values of text 101, and the accuracy was 70.3. The sensitivity was 66.7, and the specificity was 69%. This is the ROC curve of text 101. Conclusions. Although late, but the first Iraqi study from which can conclude and focus light on the following. Should be noted that signal plasma markers, text 101, ECM1, proteins are promising to be differentiated between the two types of Azoospermia and predict the success of spermatotypal, especially when complemented with testing the predictive hormone like FSH and LH, and while TEX101 seminal proteins was moderate predictive value for diagnosis of non-obstructive subtypes and the sperm retrieval rate, but unconventional alone for clinical diagnostics. From presented observation that included testicular histopathology patterns, whether of testicular extraction, sperm extraction, surgery, and seminal plasma proteins may be able to predict the chances of obtaining spermatozoa in a patient with Azoospermia, although in spite of the efficiency of some predictive procedures, no one of them are superior to the other. Recommendation from this study, the following recommendation might be proposed: increasing sample size, study other seminal plasma proteins for testicular, minimal origin and their relation to Azoospermia. More potential research regarding the correlation of seminal plasma proteins, seminal fluid, and male infertility assessment in subfertile patients. Study the seminal plasma proteins relationship with angiotensin converting enzyme and their role in regarding process diseases and sperm maturation. Comparing the seminal plasma protein assessment with other investigations like follicle-stimulating hormone, NABB, genomics, metabolomics, and other relevant investigations to differentiate between the two types of Azoospermia. Use other sperm retrieval technique and seminal plasma protein assessment. Sperm isolation technique and seminal plasma proteins assessment are compared for more accurate assessment of sperm retrieval rate. Thank you so much for joining me.